Welcome to the Arboretum. Are there a few people here who have never been here before? Good handful. Well, if you get the time afterwards, I definitely invite you to go and take a drive around the property and see some of our collections. It's a pretty neat place to work. It's 1,700 acres, so it's a nice little park that we have to ourselves here. My background is in urban forestry and plant pathology, and so the American elm to me is kind of a really important species in that the death of the American elm really instigated the interest in urban forestry and arboriculture because people started realizing how important these trees were to our everyday lives. So the question when I was asked to speak was, what topic did I want to talk about? And my one thought was, I really like how plants interact with people and how they influence people's lives. And so I thought it'd be really interesting. One of my projects that I'm working on here too is on the American elm and breeding trees that are resistant to Dutch elm disease and potentially phytoplasmas. And so since we usually don't hear about diseases that have long passed, let's do a refresher. What's happened in the last 60 years to the American elm. So Dutch elm disease is a vascular wilt disease that's caused by two different fungi, one being Ophiostoma omii and the more virulent Ophiostoma novo-omii. They're both vectored by some elm bark beetles. There's the European elm bark beetle, which is actually two different species that can vector it, and the native elm bark beetle. Here at the Arboretum, we do have a long history working with elms, but it's predominantly been with the Asian elms. Our late Dr. George Ware, who was a researcher here and the head of research, worked predominantly on Asian elms. And when working with Asian elms, two of the big features that they were looking for was the nice elm shape, which is sometimes hard to find with some of the Asian elms, and then also winter hardiness. So when you get a minute, if you guys haven't done this already, that this is the original accolade elm, which is actually just outside this door here. So when you get a chance to go take a look at it, because it's still a pretty remarkable specimen. The Arboretum has developed 3 different commercially available Asian elms varieties, one being Triumph, the accolade elm, and accommodation. So all 3 of them are excellent trees. They're available through Chicagoland Grows. And so, but my interest is more in the American elm. So when I started telling people that I was working on elms, they're like, well, hasn't the Arboretum already done elms? Like, not Americans. We need to work on Americans because they've been largely overlooked because they're sometimes a little difficult to work with. But I'm going to go back and tell you about the history of Dutch elm disease. So back in the day in France in 1917, it was first identified causing dieback of trees. In Holland in 1921, this is the first time it really got publicity, which is why it got its namesake. It was the work by Maria Beatrice Schwartz in the Netherlands who finally identified the fungus and gave it a name. And so when people talk about Dutch elm disease, they're like, oh, those Dutch, why did they bring that disease here? I'm like, Dutch elm disease is actually a complementary name to the Dutch because they've done a lot of work and they're still one of the forerunners in Dutch elm disease research. In 1927, it was identified in Britain, and then in 1933, it was first identified in the United States in Cleveland, Ohio, where they identified that it was introduced by Carpathian elm burl logs that were being introduced from France for veneer. The Britain invasion, this was kind of very telling to see how the 2 different fungal species affected trees a little bit differently and show their different virulence. The 2 elm species that they predominantly have are Ulmus carpinifolia and Ulmus glabra. In 1927, Ophiostoma omii, the less virulent strain, was introduced, and that accounted for about 10 to 20% of the dieback of the elms between then and 1960. Ophiostoma novo-omii was introduced in 1971, and in a matter of 7 years, it accounted for 70% of the elm death. So you can see that it's a much more virulent strain than the first strain, omii. It was introduced, the novo-omii was introduced into England from Canada on logs. So again, this world trade of wood products has been a problem for not just now, not with just emerald ash borer, but it's been around for a long time. And so they found that even elm trees that were resistant to olmii were susceptible to nova olmii. So that became a bigger problem when it came to breeding, because they were finally looking into breeding when olmii was a problem. By the 1930s, the American elm was the #1 street tree across the United States, which we know that because we have cities like Elmhurst, and there's always an Elm Street in every city. And when we think of traditional, America, Main Street or downtown, we think of streets lined with trees like this, because that was like the picturesque elm canopy lined street that we all love. That, I think even today we kind of want this, we want to recreate this, but in a more diverse manner. The fungus reached the Mississippi River by the 1950s, and then the Pacific coast by 1975. So here in this map, this is the native range of the American elm. So it's largely the East Coast. So the infection, it was first introduced in Ohio. So this map shows the gradual progression of the spread of the pathogen. So the red area shows the 1930s, right around Ohio. 1935 is the orange. The 40s, and then the 50s, the green area. So this is where we are, up here in the northeastern Illinois. 1965 is the dark green, and 1975 it gets to the west coast. So it did take a few years to spread, but it definitely spread pretty quickly. The disease symptoms, when you're looking for Dutch elm disease, they're pretty distinct once you have an eye for them. Sometimes it's kind of tough because they see anthracnose that sometimes has leaf curling and discoloration. But when you finally recognize that flag, it'll catch your eye, and you're just like, oh, there it is. You have this discoloration, where you can see here in this picture. It starts off as the yellowing, and the leaves will eventually curl, and they'll die, and the trees will defoliate prematurely. If the fungus is introduced into a tree by the beetle, a lot of times you see branch death, which is individual branches, one at a time, which is a little bit more of a slow, more gradual death. But if the fungus is introduced to the tree by root graft, which most trees, if they're planted within about 40 feet of each other, it's pretty much assumed that they're probably root grafted and sharing vascular systems, that the death can be a little bit faster because you have the lower part of the tree that's being affected, and you see more of a uniform canopy dieback. A lot of the symptoms you start seeing around June and July, especially July to August, they really become more prominent because of the heat of the summer. And you see that because there's the restriction of water movement, because there's the fungus moves within the vascular system and prevents water movement up into the stem. And so we're always told that the one thing, if you want to do field diagnostics, you peel back the bark and see if you have vascular streaking. Of course, too, you have to make sure that it's Dutch elm disease and not verticillium wilt you're dealing with. So again, proper diagnosis is important. So these are the symptoms you're looking for. First you get the green curling, and then they eventually turn yellow and curl. This is the flagging that I was mentioning earlier, which you can drive down the street a lot of times, especially in areas that are kind of wild, overgrown areas that have elms, and you'll see this flagging come June and July. And the next year, sometimes a lot of them are dead. The vascular streaking is pretty pronounced. The fungi are pretty easy to isolate in culture, and so it makes identification pretty easy. And then these are the European elm bark beetles. We do pheromone trapping here on the property. And so this is one year that we did pheromone trapping, and the whole trap was just riddled with elm bark beetles. Like I mentioned with the root grafting, a lot of species do root grafting. So when plants grow near each other, their roots will touch, and they'll just like think of stem girdling roots, except for in a more positive way. And they join vascular systems, and because of that, if one tree gets the disease, it will move down into the stem, across the roots, and up into the other tree, which is one reason why if you're dealing with a situation where you have multiple elm trees and one has Dutch elm disease, before you cut down the diseased tree, you want to make sure you sever that root graft. Because if you don't sever that root graft and you remove the tree, it basically becomes a sink source. Well, the remaining tree will and vacuum up the pathogen and actually will die that much faster. For localized disease spread, the root grafting is actually responsible for the majority of the death, which if you think of, if you have main streets that are completely lined with American elms, which is how it once was, if you have one tree amongst all of those that gets the disease, it just spreads down the road. And two, when the American elms were the predominant species, we didn't have the heavy pavement we do now for roads. And so, like nowadays, it's pretty much assumed that if pavement is wider than about 10 feet, not very many tree roots will grow underneath there. But at the time, they had gravel and they had brick and that sort for their roads. And so the roots did reach across the streets in that way too, so they could jump across the streets. So with host susceptibility, it was found that, lucky us, we have the most susceptible species growing here in the United States, which is the American and the red or the slippery elm. In Europe, the trees are a little bit moderately more resistant, but they're not terribly resistant. And then the highly resistant species are the Asian, Chinese, and Siberian elms. For a long time, people tried breeding American elms with some of the Asian elms to see if they can breed in some of that resistance. And they were just having no success with that at all. And that's largely because we found out that the Asian species are largely diploid, which means they have 2 copies of their chromosomes, while the American elm is tetraploid, so it has 3 copies. So just genetically, we couldn't get them to breed. Unfortunately, the tetraploid, because it has more chromosomes, naturally have larger cells. And it's this larger cell structure which leads to larger xylem vessels that make the tree more susceptible to the infection and more, because the fungus is able to move within the vascular system that much easier. So this proportion of having large vessels compared to smaller vessels that are seen in the Asian and the diploid species seems to be one key element of resistance that people are really looking into. They've also found that some of the most susceptible trees are the ones that grow really vigorously, which I actually thought that was a little counterintuitive because elms naturally have this, what we call kind of a youthful resistance. And so usually to see a tree's true resistance or how susceptible it is, you have to wait till it gets about maybe 15 years old, so about 6 inches diameter, 35 feet, 40 feet tall. Inoculate it then, because when it's a younger tree, you can easily get 6 or 8 feet of growth out of a young elm in 1 year, no problem. And with young trees, a lot of times if they get the infection, they're able to seal over that. They can grow over it so quickly that it kind of doesn't skip a beat. But once it starts getting a little bit older, slows down its growth, it's not able to seal over that infection as easily. Recently, I think it was in 2011, Alan Whitmore and Richard Olson out of the National Arboretum did work on looking at whether there were diploid American elms in the United States, which there fortunately are. So that's actually very, very exciting. I'm kind of thrilled about this. Unfortunately, up here in northern Illinois, we only have the tetraploid species. Tetraploid is indicated by the red 4s and diploid by the blue 2s. So it is kind of my hope that someday I might be able to go down south and see if I can find some diploid American elms and work on those and see how they are resistant. Because just based out of observation by Whitmore and Olson, that these diploid trees did seem to be more resistant than the tetraploid American elms. Seasonality of when the trees are infected also depend on or affect how the trees respond differently. So a spring infection, so right now is the perfect worst time to be infected because the summer, you'll start seeing symptoms in the summer, but the spring infections are actually the most harmful to the tree because the move of the spores and the mycelia throughout the vascular system are dependent on the sap movement. So right now, as the trees are leafing out, the leaves are starting to harden off, they're pulling up water exponentially almost. And so they found that it's been found that if you inoculate the tree between May, June, maybe early July, that the sap can move spores 25 to 30 feet up the stem. And so that's how it's moving. So that infection can spread that much faster. If the trees are infected in the fall, they can survive a little bit longer. And so you'll start seeing the flagging, the individual branch dieback, but they'll probably leaf out the next year. may struggle a little bit, and then you may get 2 years out of them after that, maybe. The spores are easily spread throughout the vascular system, which are introduced and then taken away from the trees by the beetles. So the beetles visit weakened or stressed elms to feed, and they usually only feed for a couple days, and then they leave and they go find a dead elm to lay their eggs. And usually these dead elms are the ones that were also infected with Dutch elm or the fungus that causes Dutch elm disease. And so it can easily move those spores back and forth because the fungus will produce thousands of these sticky spores, and the beetles will crawl through them and just be covered. Their bodies will be covered with the spores. As the beetles bore into the tree, the fungus can easily start, the spores will germinate and start growing into the vascular system, where the spores will multiply and the mycelia will start growing up and down the vascular system. And as the tree's immune response, they start producing tyloses, which are basically little balloons or air pocket bubbles, which are a way for the tree to prevent movement through the vascular system, and gums to prevent the spread up and down its system. So this here's a couple pictures. This is traits of susceptible elms. A lot of times they will produce the growth hormones to produce tyloses, which this is what the tyloses, or an individual tylose, in the vascular system. So you can see how this basically plugs the vascular system, so anything can't move up or down, which actually, if you guys remember your oak physiology, this explains one reason why white oaks are preferred for for wine and whiskey barrels, is because they're more watertight, because they naturally do this, which also makes them more resistant to oak wilt. But unfortunately, with the susceptible elms, there's this delay in response to the infection. So if you have a tree where the beetle introduces the fungus out here on the branch, the fungus keeps moving down the branch, but by the time the tree recognizes that it's infected, it tries sealing it off up here, but by the time it seals it off up here, it's already down to here. And so it's always one step behind the process. While resistant elms react much faster. They're kind of on their game. And so they also have shorter and smaller vessels, which would make them much more easy to seal off and compartmentalize. For resistant trees with breeding, it's kind of a rule of thumb that if it has less than 10% dieback a year, it's considered really good resistance. So a lot of people think of resistance as immunity, but it's not. It's just it's a lesser degree of susceptibility. So a lot of times that 10% a year is like one branch dying back, because that resistant elm was really efficient at compartmentalizing and sealing off that one branch. So as far as arboriculture goes, we usually say you never want to prune off more than 20% of the canopy a year. So if you're losing 10% a year because of a disease, still that's not that bad. It's not a concerning amount of dieback in the canopy. So this is a diagram that was recently published in 2013 by researchers in the Netherlands, because they continue to work on the disease. So on resistant trees, this is the situation that you would see, where the tree very efficiently produces tyloses that prevent the spread of these little orange spores and then the mycelia growing up through the vascular system. And so it's almost like a watertight plug that it produces. While in resistant trees, or susceptible trees, the tylose production is a little bit more spotty, or it's delayed because it's constantly trying to catch up to the infection. And the spores and the mycelia are able to make their way through that plug and then continue to move throughout the tree. There are 4 commercially available resistant American elms, and they are very good in resistance. 3 came out of the U.S. National Arboretum, which is Valley Forge, New Harmony, and Jefferson. The Princeton elm came out of Princeton Nursery in New Jersey, which the apparent tree was originally identified in 1922. Long before Dutch elm disease was even a problem here in the United States. The one interesting thing about Princeton is that it's also claimed to be phytoplasma resistant. So I don't know how true that is, because phytoplasmas are incredibly difficult to work with, which I'll get to in a minute. American elm breeding, this is a report by Smalley and Garias out of Wisconsin. Smalley did a lot of work on Dutch elm disease in Wisconsin. And so these were the kind of rough outline of his breeding program. And so you can see that it can be very arduous. And so you inoculate the trees when they're 3, 4, 7, and 9 years old. And from that, usually about 5 to 10% of the trees are considered good resistance, and so you hang on to those. So that's what's remaining of between 6,000 and 10,000 trees that you're working on. The trees that handle the inoculations, you reproduce those by grafting. And if you want to do any hybridization, you have to wait for about 12 years before they become mature enough to start producing flowers. After this, they're planted on the streets to test for hardiness, because growing in a nursery is much different from growing on roadsides, which is a pretty harsh environment. And after about 20 years and 10,000 trees, you get maybe one. That 1%, you might get one tree. Which, you guys are tree people. You know that trees don't do anything quickly. It's a labor of love. So here at the Arboretum, this is my program, so it's very much so modeled after what Smalley has done in Wisconsin. I have 4 goals to my projects with elms. One is to work on developing new resistant American elm cultivars. It's testing the currently available Dutch elm disease resistant elms for root inoculation resistance, which I'll explain that. Testing Dutch elm disease resistant elms for phytoplasma infections and how well they handle that. And then surveying for Dutch elm disease and elm phytoplasmas within the 7-county Chicago region for now. First, I have about 45 trees. I refer to them as Heimlich's elms. Gene Heimlich worked for the Illinois Natural History Survey, and he collected seeds from elm trees in Champaign-Urbana in the 1980s. I don't know how they managed to come here, but he brought the trees here, and those are where I'm working on now. And so they're about 30 years old, and those are the trees that I'm working on screening for resistance. So I'm doing the inoculation trials, propagating them, and then doing controlled crosses. When I first got here in 2009, I did inoculation trials. So I went out and inoculated each tree twice. So we have the bucket truck here. Go up, inoculate the trees in 2 different places, and then measure the streaking come that fall and then in the spring to see how well they coped with the infection. Assess the canopy symptoms. Did they have the flagging and to what degree? And then from there, I picked the top 15 trees, also based on these trees have been worked on by a previous pathologist, off of their records, what I've noticed with my inoculations, and then picked the best 15 to continue working with. Propagating elms is not an easy task. After working on elms, I would love to propagate willows for the rest of my life. Because again, labor of love. For the most part, year to year, I have about a 3% survivability rate. And so the cuttings are collected in mid-June and put in the greenhouse or put in the fog room for a couple years, and then grown in the greenhouse for a couple years. And then actually this year I'm planning on inoculating. I have enough trees that are between 2 and 5 years old that either I need to get into the ground and produce to get to the point that I can put them on streets. And I was like, oh, that's a lot of trees. I don't really want to plant that many trees. I'm like, wait, I can inoculate them, kill a bunch of them, so that'll thin them out. Job perks. So the goal is then I'll inoculate them, see how they handle through the end of the season. Whatever survive will get field grown until they're about 1-inch caliper, so about street tree planting size. And then we have a great working relationship with a lot of municipalities in this area, and they're all kind of eager to help me by taking some of these trees, planting them on really harsh environments in their municipalities, and seeing how they handle that situation. One thing that I started last year was actually collecting cuttings from local elms. So if you're familiar with the program that the U.S. Forest Service, the Northeast Research Station, has a similar program going on, which if you do Google search for survivor elm, USDA, or Northeast Research Station, you'll come up with kind of a similar program where they want people to go onto their website and log in the information for specific trees that would be a good candidate for propagation. So they look for the X and Y GPS data. They want trees of a minimum DBH, which I think are like 28 inches or 32 inches. They're nonetheless big trees, and kind of address and all that kind of stuff. So it's their intention to go around eventually and take cuttings from these trees that have survived for this long. I'm doing that on a more local scale. I've actually talked to the researchers at the Northeast Research Station and told them what I was doing with Heimlich's elms and then what I'm doing here. And they're like, oh wow, you're 20 years ahead of us. Keep going, we'll catch up. I was like, okay, great. There's more people out there working on American elms, which is very encouraging. So last year was the first year I did this. Again, I worked with local municipal foresters, and which they're all kind of, they're fun to work with because they've been in the industry for a long time and they have to deal with homeowners a lot, and so they get a little grumpy. And then, so I go to them, it's like, hey, do you have any American elms in your municipality that you would love to see propagated? And you see this twinkle in their eye, they're like, yes! And so on average, I try to sample from at least 9 trees a year because, again, with a 3% success rate, we have to do a lot of propagations or a lot of cuttings to do this. So I'm looking for trees that are either, they have been untreated for 5-10 years at least, or have never been treated. And because these trees have been living in that urban environment for so long, it can kind of be assumed that they are pretty tolerant to the harsh urban environment, the compacted soil, salt, water, flooding, and unavailability and pollution. And so that program I'm actually really interested in because, one, they're local trees, so people hopefully in the Chicagoland area will be more motivated to show more interest on these trees because they are kind of the hometown trees. The controlled crosses I'm working on are, again, with the Heimlich's elms. And so these are the parent trees out here in Ware Field, which is kind of the research area, just over that way. But they're growing, so our tree crews helped me quite a bit with doing this, with bagging them, and then going out there with little paintbrushes and pollinating for me. I told them I wanted to get them little bumblebee wings, and they can just, they weren't into that. But doing controlled crosses, so those crosses are about 2 or 3 years old right now, so I have plans on inoculating them to see how well they cope, if they're as good as their parents. The root inoculation is something that's a little bit new to the elm breeding world, from what I found in the literature, because one of the problems, which I showed earlier, that 60 to 80% of the local spread is because of root grafting. And so if elms are becoming popular again, there's that concern that if we plant elms next to elms next to elms, are we going to have the root grafting problem? Because most of the resistance breeding looks at whether the tree was inoculated by a beetle, by the canopy introduction. Nobody's looked at, well, how do they respond to a root introduction? So we could have these fantastic trees, but if they root graft, are they as susceptible as the old wild type susceptible American elms? So I've done 2 projects so far. One is ongoing. One was just kind of hey, let's see how it works. We had some duplicates of the Heimlich's elms that I wanted to see how they responded. And so actually air spaded a little bit and then found roots and inoculated the trees that way. And then, and this was in a planting, they're all American elms planted in one I think it was like 8-foot blocks. And they were there for probably 10 or 15 years. So I watched the surrounding trees too to see if they were root grafted and if it moved. The other is working on the Princeton Valley Forge, Jefferson, and New Harmony elms, and deliberately trying to get them to root graft and then growing them in the same pot. And the plan is, once I can confirm that they're root grafted, is to inoculate one and see how the other one responds. So it's great that we have trees that are resistant to Dutch elm disease. Hopefully we can bring back the American elm. All well is good. Unfortunately, just because they're Dutch elm disease resistant doesn't mean they're resistant to another pathogen, which are phytoplasmas. If you're familiar with elm yellows, there's the cloverleaf proliferation phytoplasma, the elm phloem necrosis phytoplasma. And after talking to Gary Moorman, who's out of Penn State, they have different strains of phytoplasmas by state and region, and it's kind of a mess. So phytoplasmas, for the most part in elms, have very similar symptoms as Dutch elm disease. So a lot of times when municipalities or anybody sees a dying elm that has the canopy flagging, a thin canopy, and then a vascular discoloration, not necessarily streaking, they automatically say, oh, it's Dutch elm disease, remove it. Which really the prognosis is the same between phytoplasmas and Dutch elm disease. The tree's going to die. The problem is that there's a concern of, if we're going to bring back the elm, knowing where there's pockets of phytoplasma versus Dutch elm disease will definitely help with putting the right plant in the right place. So a little information on phytoplasmas. Phytoplasmas are almost ridiculous to work with, which is why I'm working with them, which is fun. Sometimes for a long time they were considered fastidious bacteria. So to be fastidious, you're very particular. You want to grow in a very particular environment, which makes them a little bit more difficult because you cannot grow them in culture. You cannot move the pathogen easily by making a pruning cut in one tree and making a pruning cut on another tree. You can't vector them that way, which is kind of nice in a way. You don't have to worry about that. They're vectored by insects, by sap-sucking insects, but even insects cannot feed on one tree and immediately spread it within that tree or to another tree. There's a gestation period within the guts of the insects. So there's usually between like a 1 to 3 week gestation period before even the insect can move the pathogen. They also overwinter in the roots, and so you have to wait until later in the growing season before the phytoplasmas are actually moved around within the canopy of the tree. And so you can see the problems that I'm running up into. So again, using the Dutch elm disease resistant American elms and looking to see how well they cope with this, or actually first seeing if I can inoculate them. So what I've been doing is I have a tree that I know has an elm yellow, or an elm phytoplasma. I don't know exactly which phytoplasma it is because that identification takes a little bit more, but for now it's just a phytoplasma. I just want to work with it. So what I've been doing is taking branch cuttings and actually trying to graft vascular tissue from one tree onto another. So just like you would graft an apple tree or anything like that, where you take a chunk of tissue and try to graft it into the other one. So I've been doing this for about 2 or 3 years. I'm going to continue doing it and seeing how it works. I'm sure I'll let you all know. So the other project with phytoplasmas is actually surveying for the phytoplasma and for Dutch elm disease. So again, working with municipal foresters, park districts, all those. And really I want to be able to make a map of the Chicago region for now, looking at, okay, where is there Dutch elm disease and where are there phytoplasmas? Because for the most part, when, again, an elm's dying, people say, oh, it's Dutch elm disease, remove it, no big deal. But this is one reason why proper diagnosis, I'm a diagnostician, proper diagnosis is always important. You should always go take your samples to a lab. But having it properly diagnosed is really important because if you have a pocket or an area that Dutch elm disease is prevalent, you can plant the Dutch elm disease resistant elms. Not a problem. It should grow fine. But if you have an area that you say has Dutch elm disease, but really they're phytoplasma, even if you plant those resistant elms, they're going to die. So it would be kind of nice to know whether you can plant elms in an area or maybe you should just avoid them and plant some other things. Fortunately, sampling and doing the surveying isn't terribly difficult. Testing for Dutch elm disease, to do confirmation testing, you have to do culturing. Ophiostoma is very easy to grow out in culture. It takes about a week, 2 weeks, and then you can have your answer. If it's negative for Dutch elm disease, you start looking to phytoplasmas. Fortunately, phytoplasmas are extremely stable, and so you can collect the tissue, put it in the freezer, and hang on to it for years before you need to run the test or you have enough to run the test, because doing the test is actually a real-time PCR test. Gary Morman published some protocols for doing the testing that way. So with the elm projects, that's pretty much what I've been working on here and the history or what has come in of elms since the Dutch elm disease come to the U.S. I've had a great deal of help with this project, which I'm always very thankful. So for the elm part of my talk, do you guys have any questions? No? Okay.
Question. Are you still looking for resistant elms?
Absolutely.
In the community?
Yeah.
I need to talk to you.
Okay. I will talk to anybody about elms. Two, I have my weaknesses, disease, trees, you know. If you want some really good information on the history of Dutch elm disease and how it's spread around the world and different species and all that sort, and how trees respond to the pathogen, I find host-pathogen interactions is fascinating. It's kind of like chemical warfare on a very small level. Here's some really good sources of information that I use for this presentation. The next pathogen I'm talking about causes a disease that we often refer to as sudden oak death. But before I get into that, I kind of want to give you guys background information on Phytophthora species, because I'm sure you guys haven't read about Phytophthora species in a while. Does anybody read? No? Okay. Losers. So if you remember your American history, where the Irish potato famine and all the Irish came to the United States in the late 1800s, do you guys remember that? Remember hearing about that? Some of us are a product of that. So the late blight of potato is caused by a Phytophthora species. It's caused by Phytophthora infestans. And Phytophthora, the name itself, although a scientific name, is actually a Greek name. Phyto meaning plant, Tophthora meaning destroyer. They're very good at what they do. There's about at least 123 different species around the world that cause diseases on herbaceous plants, woody plants, forest plants, commodities, whole gamut of different plants. Pretty much if it's a plant, it probably has a Phytophthora species. They're not true fungi. They're more closely related to golden brown algae. So a lot of times people refer to them as fungal-like organisms. But since I was kind of trained in the old school way, if I call them a fungus, please forgive me, because it happens. And so again, identification is important when you're dealing with a root rot or canker or anything else like that, in that if you're going to use any kind of chemical controls, having that proper identification is important, because being a fungal-like organism, you cannot use the same fungicides as if you were working with a basidiomycete, ascomycete, or deuteromycete. So you can apply as many gallons as you want. Actually, you shouldn't go by the label. But having that properly identified and applying the proper chemical and the right rate, you'll have better control. So Phytophthora species are soil and water inhabitants predominantly. Again, they're water molds. They can survive in soil for years even without a host. There was some really interesting research on Phytophthora cinnamomi in Australia that was causing dieback of eucalyptus trees that found that Phytophthora cinnamomi can survive in barren soil for 20 years and still germinate and be viable. No problem. And so again, my background in my master's degree was looking at Phytophthora Ramorum in Georgia, and I really learned that you do not want any kind of soil-borne pathogens. They are just a pain in the tuchus to deal with, and just try to prevent them and avoid them as much as possible. So these pathogens definitely like cool, wet conditions. So some background on the different spores. They produce 4 different types of spores. Up here are the chlamydospores, which are asexual spores, and this is the oospore with the antheridium. And these 2 types of spores are used for long-term survival, so they can lay dormant for several years. So it's the chlamydospores with Phytophthora cinnamomi in Australia that could survive for 20 years. During the growing season and when the environment's favorable, Phytophthora species will produce sporangia, which they can either directly germinate and infect the host plant, or if there's ample free water, they can produce zoospores, which are seen down here. So this is the sporangia. So the zoospores they'll pop and they'll start swimming out of the sporangium. And these zoospores are even chemotaxic. That means they're drawn towards favorable chemicals. So if you have a host plant's roots in the water, especially if they're damaged, the zoospores are actually attracted to those and they'll swim to those roots. This is the disease cycle of Phytophthora infestans, just kind of giving you an idea of how all these spores work. So you have the infected plant here, and in some situations they'll produce the sexual spores, which are the oospore, or the oogonium and the antheridium, which produce the oospore, which I showed you on the last slide. They can germinate and produce the sporangium, and the sporangium can either germinate directly and infect the plant, or can produce these zoospores, which again can swim and produce and infect the plant. With Phytophthora Warmnow, there's sometimes some confusion when talking to people because actually it's one pathogen, but it causes two different diseases depending on which host plant you're looking at. So sometimes it's referred to as sudden oak death, and sometimes it's referred to as worm blight. We pathologists tend to do that. You put Xylella fastidiosa on a shade tree, it's bacterial leaf scorch. You put Xylella fastidiosa on a grape, it's Pierce's disease. So knowing what is actually causing the disease is also important. Phytophthora Ramorum was first identified in California in the 1990s, and it was then that people started really noticing that tan oaks and oak species were starting to die, which tan oaks, although also in the Fagaceae, they're actually a lithocarpus, not a quercus species, but just widespread dieback of these trees. And finally, in 2001, a name was put to the organism. We knew it was a Phytophthora species, but had never seen it before. It was given the name Phytophthora Ramorum, and unfortunately also in 2001, it was detected in California nurseries. And this is kind of where the story starts with the United States and the spread of Phytophthora Ramorum. Because in 2004, camellia and rhododendron plants were shipped to, I think it was 38 states across the United States and into British Columbia. And so with my story, this is where I started because that's when I started graduate school at the University of Georgia, and the state of Georgia received 28,000 potentially infected camellia plants. 8,000 of those went to the largest production nursery in Georgia, and so as soon as they heard wind of this, they put it in their fire pits and they torched them and wanted nothing to do with it because it's a federally quarantined pathogen. So then there's only 20,000 potentially infected camellias in the state of Georgia. A homeowner came forward and says, oh, I think I have one, and sure enough, it had Phytophthora Ramorum. So then there's only 19,999 potentially infected camellias in the state of Georgia. So my project looked at trying to identifying a kind of a baseline species of what Phytophthora species naturally exist in Georgia, and then also seeing if I can find Phytophthora Ramorum in nurseries, in streams that are adjacent to nurseries, and then even in the kind of foothill regions, because a lot of people have second houses where they want a landscape, and so they'll buy plants and plant them out there. So in the United States, at this point, the biggest, or the area that has consistently been infested are 14 coastal counties, nurseries in these counties in California, and one county in Oregon. Because of this, pretty much any kind of plant material, soils that come out of this area are under quarantine and have to be inspected to make sure they're free of the pathogen. The rest of the country, for the most part, Phytophthora has been found in nurseries, in soils, and in natural waterways. And especially in the nurseries, these come in the form of the trace-forward samples. So trace-forward samples are, I think this is an APHIS term, where if a nursery ships out plants, so nursery A ships plants to nursery B, then nursery A is found to be infested with Phytophthora Ramorum. They'll open their books and see who they shipped to, and they'll go to all those receiving nurseries and inspect those plants to see if it got moved. So those are trace-forward samples. Also in these nurseries that received trace-forward positive plants, they will also do water sampling and stream baiting. As a way for water conservation, which is actually a really good idea, and water pollution, a lot of nurseries use recirculated irrigation water. And so all the runoff that comes from the plants goes into a retention pond that gets reused, which as far as water conservation is an excellent way to manage that problem. Unfortunately for pathology, it's an excellent way to move pathogens in a very small area very, very quickly. So the Phytophthora Ramorum now has been found almost worldwide, Canada, the United Kingdom, and then 21 other countries in Europe. It's predominantly been found on rhododendron and viburnum in nurseries, and that seems to be the major source of spread across the world. And in Europe, it's also been found in parks, gardens, woodlands, and forests. It seems to have spread very, very quickly. This map, I'm not going to go over all the fine details, but it just shows that a lot of these countries in Europe are being very proactive in trying to detect the pathogen, eradicate the pathogen. So all these different colors indicate whether it's been found in a nursery and eradicated the nursery, if it's been found in a garden but not in a nursery. There's all these different combinations. So nonetheless, Europe is on the ball, and they're doing an excellent job trying to quarantine and eradicate the pathogen. So unlike Dutch elm disease, which seems, when working with Phytophthora Ramorum, Dutch elm disease, although it was devastating, seems pretty easy because it has largely 1 or 2, maybe a dozen different hosts. Phytophthora Ramorum has at least 45 proven plant host species and 91 associated plant host species. So the difference between the 45 and the 91 is whether Koch's postulates has been completed on it. So Koch's postulates is a basic principle in all types of pathology that proves that one particular pathogen causes one particular disease on one particular host. And so it does take time to prove that whole association. So the 45 are proven to be hosts. The 91, they're still working on Koch's postulates to complete that process. This list is published by APHIS and it's updated pretty frequently. And so you can go and see that the host list for Phytophthora Warmnow is extensive. Forest plants, shade plants, native trees and shrubs, herbaceous plants, everything pretty much across the board. It can be potentially a host plant. So the 1 pathogen, 2 diseases. First I'm going to go over sudden oak death, which is neither sudden nor does it only affect oaks. I don't even know why. But on the host plants, on the trees, so this sudden oak death occurs on the large overstory shade trees or forest trees. And on these trees, it causes a canker disease. So it's a kind of a typical fungal-like canker where eventually it'll girdle the tree and kill the tree that way. These trees are a lot of times killed over, usually it's between 3 or more years. But the interesting fact with sudden oak death is that on these overstory hosts, that the fungus does not produce spores. So in that fashion, it's actually pretty good because although the tree dies, the Phytophthora species is trapped. It doesn't move unless you move the wood chips or make a pruning cut and go prune something else. So it produces the cankers on the stems, which restricts the water movement. A lot of times on the stems you see an orange-brown ooze coming from the canker, which is also common with other Phytophthora cankers. So just because you see an oozing canker on an oak tree, don't panic. It may be a Phytophthora species, but that's not the Phytophthora species. With a lot of diebacks, you get the pale green color that eventually turn brown and they'll die. The leaves will drop prematurely and you'll start seeing a thinning canopy. But there are a lot of lookalikes. I mean, what I just described describes a whole gamut of different diseases and insect problems on shade trees. So again, proper identification is always important because it can look like oak wilt, oak decline, other Phytophthora species, oak borers, two-line chestnut borer, this describes them all. So make sure you get your diseases and insects properly identified. So the trees can live between 1 to 5 years with the cankers. Some tan oaks have found to be a little bit resistant, and even some tan oaks that were previously infected have seemed to be recovering, so there's kind of hope in that fashion. The cankers I was describing here, you can see that the tan ooze that goes down the stem, and then if you peel off the bark, probably right up in this area, you'll start seeing these very dark cankers with a nice dark margin. A lot of times these trees, because they're stressed by a primary pathogen, a lot of secondary things will come moving in. These are usually the opportunistic insects and pathogens like hypoxylin canker, which is up here, and then ambrosia and different bark beetles will start infesting the tree. So the impact of this pathogen on the world's forests has been pretty extensive. So on the top is the impact of Japanese larch in southwestern England. So you can see a lot of the brown and dying tips of the trees here. In California, tan oaks and oaks on the California hillside have been greatly impacted. With Ramorum blight, this is the disease that is more of a foliar disease, and a lot of times it's more on understory plants. They're sometimes killed, but oftentimes they're not. And it's on these hosts is when the spores are produced, and this is how it's really been spread. Especially when you're talking about nursery stock, you think about it, that a lot of times people will buy rhododendrons and azaleas and they'll want to plant them in the shade of their oak tree. So you can see how it can spread very easily that way. For the most part, on these foliar hosts, you have the angular leaf or irregular leaf spots. And a lot of times, like a lot of different foliar pathogens, is that the leaves a lot of times hang down. And so the infection sometimes will start at the tips of the leaves on the margin, because that's where water accumulates in the morning or when the dew. It can grow eventually down the petiole and into the host plant and causing a blight. So a blight is wilt plus death, so you get that shepherd's crook, and then you'll get premature leaf drop of those symptomatic leaves. So the leaf spots here, this was a water bait that was positive for Phytophthora Ramorum, but you can see the leaf spots are not perfectly round, and they're not angular, they're not limited by the veins. They do have kind of this feathery appearance. And then the non-leaf baits, this is from a nursery where you can see the leaf spots. This one starting at the tip, and this is the base of the leaves. The tip blight, so the tips of the plants can be infected, and eventually they will blight, and you'll sometimes see these perfect shepherd's crooks. Spread within the nurseries has been a major problem, but it's also been an excellent way to prevent the spread. Nurseries have been very cooperative with APHIS in trying to quarantine this pathogen. Within the nurseries, you have plants sitting next to other plants, so you get contamination that way. You have contaminated irrigation water. It can also reside in the soil. So you'll have the leaves, the infected leaves, drop to the soil. And although for the most part in nurseries, they're gravel beds, but over time, all sorts of soilless mixtures and leaves and everything spill out. So you have somewhat of a soil ground to the nursery beds. So these trace forward samples are really what APHIS has been following after inspecting nurseries. Nurseries, after they're found to be positive or have positive plants, they are monitored for several years, and they take water and soil and water samples. In 2010, Illinois, there was one nursery in Illinois and 2 in Iowa that did receive trace forward positive samples. And at that time, I was actually contacted by Steve Oak from the Forest Service, because the Forest Service was starting to look at surveying forests or natural areas around these nurseries to see if it's leaving the nurseries because of these trace forward samples. So just kind of give you an idea of some of the nurseries that I worked with. These are both in Georgia. This retail nursery, when they get their nursery stock, actually just stock their plants up right around this is their irrigation pond. This is the largest nursery in Georgia, which is a couple hundred acres, and they have 13 recirculated irrigation ponds that are all tied in with each other, and so they kind of go in one big cycle. And so when they pump out of these streams, it can come from any number of blocks of plants with different types of problems. The APHIS is looking at both host plants and non-host plants, because just because it's a non-host, if it was irrigated with infested water, or if it was sitting next to an area or a plant that was infested, it can easily move on even containers. These regulations have drastically reduced the amount of Phytophthora Warmnow in nursery stock, and it's actually been pretty remarkable. So any nursery stock coming out of California, Oregon, and Washington are largely regulated and monitored very closely. One of the problems they had is that a lot of times people will go into the forested areas and dig up plants and take things that way, and that's another way people are spreading this pathogen. Sometimes it's tourists, sometimes it's nursery employees that just want to get new breeding stock. And then surveying plants that are irrigated by potentially infested water. So the high-risk species, it's been kind of quoted, these are kind of the 5 horsemen of the apocalypse, I think Steve said, because on these 5 species, Camellia rhododendron, Viburnum pieris, and Kalmia, the pathogen has most commonly been reported on them, so they're extremely susceptible. And on all these 5 species, the fungus produces an abundance of spores. And so they can move the pathogen very, very quickly. So the proposed movement within the nurseries, first you have one plant that comes in that has some disease, which most plants, they get disease, and so a lot of times we kind of dismiss it and keep moving on and use the plant. So the plant can be put in with the rest of the plants and spread locally that way. You have water runoff that can come off these plants if the plant's used for propagation with the cuttings, and then if the plant dies or the dead material falls to the ground, it gets put into the coal piles. which again, a lot of runoff from nurseries goes into either adjacent streams or into their recirculated irrigation water. Between 2003 and 2011, 464 nurseries were tested positive for having either trace forward positive samples or positive samples, and this is in 27 different states. 2012, the number went down, obviously individually, to 21 nurseries, and 2013 and 16 nurseries. So you can see that these regulations are being very effective. Most of these nurseries in the 464 received those plants, so they are trace forward samples. This is a Quonset in a Georgia nursery that was tested positive for Phytophthora Warmnow. Where on this side you have Camellia, and on this side you have Azalea. So you have two of the best species right next to each other. The nursery survey in 2004 reported 18 nurseries, and 8 of those ship interstate. So one thing with federal regulation is that if your nursery stock doesn't leave the state, it's not as heavily regulated. And so some of these nurseries that have been tested positive only ship within that state, so they're not watched as much. So these are the states that reported having positive samples in 2014. So some of my work in Georgia, just to kind of illustrate how it can spread very quickly within the nursery. This is the Quonset that's in Alpharetta Nursery outside of Atlanta that was tested positive for having Phytophthora Warmnow. So when I was talking about taking soil baits, the soil is actually collected from the ground and flooded with water, and 2 different host plant species leaf samples are floated on top of the water for usually 42 to 72 hours, or 48 to 72 hours, and then plated out. So again, those zoospores are released when there's a lot of free water, and they can swim towards the host plant tissue, so it makes baiting a little bit easier. And then we also collected plant tissue that fell, showing symptoms on the ground, and plated those out for confirmation. So this is a nice storytelling illustration of how easily it can move. So this house here, house 1, is actually this Quonset, or the shade house. So once some of the plants were found to be positive for Phytophthora Warmnow, we went in and I just divided up the house into 5 different areas for the sake of sampling, and went through and took soils and water baited them. So in May 2005 is when I collected the samples, and all 5 of those samples, out of all of those, only samples out of section 3 were positive for Phytophthora Warmnow. But have you guys ever been in Georgia in May? It's really hot. And so because Phytophthora species are cool, moist, they prefer cool, moist conditions, we had the samples refrigerated for a period of time, and then in September went back and rebaited these samples and retested these samples. And at that time, all 5 areas were positive for Phytophthora Warmnow. They just needed that cooling dormancy period. In October, so what happens then is that this entire shade house has been quarantined. No plants can be stored in there. People are not supposed to go in there. And so the nursery owners took all the plants, which you saw there was a lot. They took all those plants, which are also quarantined, and they cannot be sold. They have to hang on to those plants. They took all those plants and they moved into house 2. So in October of 2005, since they moved those plants, we sampled from not only the 5 original spaces, but also from the next area, which again, because they moved all those plants, they also moved the pathogen, and that house was then also quarantined. In November, resampled, and although this house was negative, the one new development was that there was this drainage area that both these houses drained down to, and then there was a nursery, the irrigation pond down here, and during periods of flood, it would actually flood into a neighboring stream. In May of 2006, so we found that it did overwinter in Georgia. This house has been empty for quite a few months, but it's still that original Area 3 was positive. House 2 was still positive. And at that time, some of these plants had gotten quite tall, and so they moved some of the bigger containers outside of the house. And so just out of curiosity, I just tipped the plant over and just scraped the soil off the bottom of the container and was able to isolate Phytophthora Warmnow from the bottom of the containers. So just by moving the containers and even walking in between the 2 houses, that was all then contaminated with Phytophthora Ramorum. So this is the neighboring stream. So if their irrigation pond gets low, they actually pump out of the stream and into their irrigation pond. But during periods of flooding, it would drain into another stream that goes into a residential area. Phytophthora Warmnow has been found in different waterways in different states. Sometimes this changes here and there, but especially states in the southeast have been especially a problem, largely because of the understory of their forests. Mississippi, Pennsylvania, and Florida have largely been found to be positive, and there's also been New York. I don't have more recent waterway baiting data. 2013, 12 states and 104 streams were surveyed. And this included the Illinois, Iowa surveys that we did, which were both negative for Phytophthora Warmnow, but you can see, well, these are just the numbers that were surveyed, not that were positive. So this is the Iowa nursery that received infested plants. So you can see the greenhouse right here, and all that runoff comes into this waterway that leaves the area. So when you do water baiting, you take leaf samples, so we use rhododendron leaves, and put them into these cages and floated them into the stream for between 24 and 72 hours, and then retrieve them, and you can see the little the spots on the leaves. You take samples from those and then bait them out and test them for Phytophthora Warmnow. So far, I've largely just covered the impact and spread within the nurseries, but even, especially in the southeast, there is huge ecological threats of Phytophthora Warmnow getting out into the environment. Again, has anybody ever spent time in southeastern United States and seen the understory? This is one of the forests I surveyed at, and all this, the understory, is all rhododendron. I mean, the rhododendron, camellia, those are their native, that's their buckthorn and honeysuckle, except for it's in a good way. So you have this loss of habitat, you'll get soil erosion, and there's also the problems with change of fire frequency and intensity. One of the great parts of Phytophthora Warmnow is that the redwoods are not a host to Phytophthora Warmnow, which is excellent because that would be a huge loss. Unfortunately, I recently read that because all these oaks and tan oaks in California have been dying, the fires, when they do have fires, are burning more intensely and more frequently, and so the redwoods are actually being killed because of the forest fires and not the pathogen. So there is kind of unseen consequences from this. The Forest Service and APHIS and many different organizations got together, and they put together this map looking at what are the highest risk areas in the United States for Phytophthora establishing. So the green areas are of low risk. These standards were all based on average rain, humidity, temperatures, understory plants, all these variables all put together. And so up here in the upper Midwest, there's pretty much a low risk. Southern Illinois has a moderate risk, which is the orange, and then the high risk areas are the red. So these are the areas that have the nice amount of rain and proper understory or host plants. In the eastern United States, there is that threat of, if it does get out, there is the environmental threat because there are quite a few native species that could be affected by this pathogen. Also, just because sudden oak death and oaks are a host species to it, I looked up this Forest Service map of oak populations, and so definitely, there's the concern that although it won't help spread the pathogen, it can definitely kill quite a few trees if it's allowed to get out into the environment. There are fungicides available. I'm not going to get into the chemical management of this, but there are several chemicals that are labeled for this. This is a great resource for that information if you're interested in it. For the most part, a lot of the tests largely looked at rhododendron, camellia, pieris, and viburnum. So I don't know how the host list is beyond that. But a lot of times the chemical management is largely preventative than curative, which is true for a lot of plant diseases. So to identify a pathogen, really you need to culture it out or do a DNA test. But you have to remember that it does look like other problems. So what do you do if you think you might have a Phytophthora Warmnow infected plant? Whether you're a homeowner, you bought some plants, or you're a nursery inspector, you're a municipal employee, first, don't panic. But contact your state's extension plant clinic. They're really the go-to resource in this area. So even if we have samples that are extremely suspicious, we send them to the University of Illinois. Illinois sends them to our regional lab, which is Michigan, and they can send it to Beltsville, Maryland, which is the national lab, and they'll test it for Phytophthora Warmnow. So it's not until the results come out of Beltsville, Maryland, that we can actually say it's confirmed positive. Until then, it's suspect, but really, you're just not supposed to talk about it. So what can you do if you're interested in any insect and disease problems, not just this? We're going to be talking about a lot of invasives today. Scouting for samples. Here at the Arboretum, I have a small army of volunteers that help me scout for insect and disease problems, which we report. Don't guess, and you want to have problems properly identified. It was interesting, this past Saturday, I taught a class on kind of an intro to diagnostics called What's Wrong with My Plant through the education department here. And so I covered insects, diseases, and abiotics in 3 hours. It was interesting. And one of the best feedback I got from one of my students who kind of came in, I kind of got the feeling that he thought he knew everything already. And after the class, I'm just like, so what do you think of it? He's like, wow, diagnostics, like, it's way more involved than I thought it was. It's like, yeah. Because a lot of it's like, okay, you have this plant, is it, A, B, or C? And so you have to rule out all these things. So make sure if you do collect samples for a lab, they're good samples, which all extension plant clinics have information on how to collect good samples. If you're suspicious, quarantine plants, and make sure you sanitize your pruning tools. I can't emphasize that enough. Tool sanitation can be the best way. 20% of alcohol can save an entire tree. And rubbing alcohol works perfectly fine, or even Lysol. So when you do receive plants that look a little suspicious, don't use them. Make sure you just set them aside and wait for them to be properly diagnosed. A lot of times too, a lot of people don't, when they go to the nurseries, they don't pull plants out of containers to really look at their root systems. I guess I'm a root and soil pathogen person, so that's one of the first things I do, which nurseries kind of hate this, but you don't also want to take something home and contaminate your soil and have to deal with that for the rest of your life. So if you have a suspicious plant, set it aside. Any of these, types of quarantine or preventative are still cheaper than any chemical management you can ever apply. A lot of times too, nurseries will spray pesticides to prevent disease, and some of these things won't come to light until you've had it for maybe a couple weeks. So we've all been down that road that you buy a couple of plants to put in your yard, and they just manage to sit there for a few weeks before you get the time to actually put them in the ground. And in that time, you can start seeing these symptoms. So again, at that time, put them in quarantine and have them properly identified. Soil sanitation is an incredibly difficult thing to do. For the most part, it's just easier to prevent any kind of soil movement of infected plant tissue, because for the most part, and this goes true for Phytophthora Warmnow, only soil sterilants or soil fumigants can be used to prevent that in the soil. So the nursery that I showed you the move in, the Ramorum's movement within the nursery, because that's all gravel beds. And so for soil fumigants, which I think they're either using Vapam or methylbromide, I think it's Vapam, but nonetheless, it's a gas that's injected into the soil. They have prongs 12 to 16 inches, and they actually have to get it down into the soil and then fumigate it that way, and then they tarp it. Well, because that's gravel, they weren't able to do that, so they ended up just paving the entire area. So basically, the pathogen is just sealed in. And even the nursery in California that spread the pathogen, that shipped the pathogen to many different states, that nursery is now gone. It's actually now, I've been told, a housing division, which part of me thinks it's funny because good luck landscaping. But don't replant infested areas with the host plants. So again, proper identification of the pathogen. If you know you have verticillium or Phytophthora in an area, find out what's a host plant and just don't plant that. It's not a lost cause. You can still work with that area. But fungicide drench treatments are usually pretty expensive, and it's kind of hit and miss whether they're successful. If you're interested in reading more on the chronology of the movement and spread of Phytophthora Ramorum within the United States and other information on the background of Phytophthora Ramorum, these are excellent resources on the suddenoakdeath.org website and then the APHIS website. A lot of this information came from the APHIS website, Forest Service, and different state and regional plant clinics. Here at the Arboretum, I mentioned earlier that I do insect and disease scouting, and we do report it. One way we report it is through the Plant Healthcare Report, which is published throughout the growing season. It is available through our website, which is here. Even if you go to our website and type in Plant Healthcare Report, it will come up. The Plant Healthcare Report also has a Facebook page, because right now the report's being published bi-weekly, and so if anything new and urgent comes up, I will immediately post it to the Facebook page. Like when elm bark beetles are found in the pheromone traps, I'll post it, or ash lilac borers are found in the pheromone traps, I'll post that. And we do the first report of any insect and disease problems, so people, if they're going to apply management tactics, they can apply them in an efficient manner. I too have a Facebook page, which if you want to follow me, I post a lot of things on insects, disease, gross, squishy, disgusting blobs of slime, that kind of things, science and that sort. So you're welcome to follow me or friend me. If you have any questions, you're more than welcome to email me. I am always looking for good elm samples. I'm getting to the point that I don't know how far I'm able to travel outside the Chicago region right now, but if you have any questions or even disease questions, you're welcome to contact me for any help. But I will say the University of Illinois has an excellent extension plant clinic, and they are also an excellent resource of information.